Okay, so what have you heard about today? You've heard about DNA identification and this pathway that can be followed when DNA identification is done. In the simplest case, when there's only one individual in the biological sample and there's a lot of DNA from that person, then there's essentially no uncertainty. That individual's DNA is worked up like a reference sample. If you want to build a DNA database or a reference sample in a case with a buccal swab, it used to be with blood. Very simple data is generated where at each locus there's only one or two peaks. This is obviously a 10-12 because there's no uncertainty. There may have been 100 possibilities before you looked at the data. In the population, maybe 5% of people would have a 10-12 and 5% would have an 11-12. But based on this data, there's only one thing you can really infer if the data is reliable. This collapses all of that probability to just one possibility, and that's an evidence genotype or reference genotype of 10-12. The inference is really easy when it's not a mixture and there's a lot of DNA. And that's what you're used to, and that's what happens on TV. But it happens a lot less in the real world right now. Then when you make a comparison, you can get a perfect match statistic up in the gazillions. Okay. But with mixtures, there are human interpretation issues. The data may be very good and very informative, but if people don't have methods or calculators and machines to help them interpret it, they can call it inconclusive. If the peaks are too low because there are these artificial thresholds, and then in 2010 the thresholds go up 3 times higher, so you can't even get there, then they can't interpret the data under those rules. There may be too many contributors for them to handle. We've done studies with some labs where after they've seen how people have interpreted 3-person mixtures, they've stopped the practice in their lab. They don't think it works or that it's reliable. 4 or 5 contributors didn't make much of a difference in the study because the analysts got no results anyway, even though the computer did. And this is becoming published in the literature more and more, the potential for examination bias. What happens if a person peeks at the data before they make a comparison? The reason subjectivity can come in is that in the protocols that people follow, they can determine that a particular locus's data is unreliable or there's dropout, and a subjective decision can get made. And they may not think of it as subjective, but other scientists might, and a court might. And what happens then with DNA mixtures is that the DNA pathway is broken. Wonderful evidence is produced which you might think should be a 7-14 and a 10-12. In your mind, you could separate out the genotype, but the human protocols that people follow generally don't allow for it. And they can't infer anything, so the pathway is broken. No genotype is produced, and no comparison is made, and perfectly informative data that could be used for exonerations or for identifying criminals or for anything you'd need DNA evidence for is just thrown out. TrueAllele Casework was the first of a technology. There are other computers like this coming into being. That's goal is to preserve DNA information. It uses all the peaks, high or low. If you have a rich enough statistical model with hundreds of variables, you model the low peaks as well as the high peaks. You model the noise. There is no threshold. In fact, you model how much variation there is in each peak. You customize a probability distribution to each peak based on what the data is trying to tell you. It can handle any number of contributors. We've validated up to 5, but the technology goes up to any number. And it's completely objective. There's no chance of bias. How can it be biased if it never saw what the answer was supposed to be? And it's documented that in how it ran. And so with excellent crime lab data, like we have in Allegheny County, and a good genotyping science with TrueAllele, the DNA pathway is restored. Mixtures can be interpreted. We just give up the hope that there would be only one value, that it would only be a 10-10. It would be that easy for the lab, that easy for an analyst, and resolve ourselves to saying, well, it's probability. That's life. Fortunately, all the math works, including match statistics. And we can interpret these mixtures, get genotypes, make comparisons, and produce match statistics that are useful for criminal justice. Now, there's a problem, and it's, not just in Allegheny County, it's around the country and around the world. We made some estimates. You remember that number that I asked you to remember, 375? Right, from the number of cases reported in the Washington Post that were sent back by the lab to the Attorney General saying we can't interpret them before they brought a computer in. I'm just going to give you two rough estimates. This is the state of Virginia, 375 cases, roughly in the year after Swigdam 2010 came out. Could have been a larger period of time, could have been smaller. But in the last 4 years, 375 times 4 is 1,500. That's 1,500 cases in a place with a population like Virginia. Virginia's population is about 8 million people. In the US, there are 320 million people. So if you want to extrapolate, dividing 320 by 8 is a factor of 40 if you want to go from Virginia to the whole country. So if you multiply the Virginia experience of getting inconclusive results on 1,500 cases, but a factor of 40, that's about 60,000 inconclusive mixtures. New York, which is about to go live with TrueAllele, is also counting. In a recent week, they saw that out of 40 cases, 20 of them were deemed inconclusive, even though the analyst looking at them said, well, these are perfectly fine for TrueAllele. New York has trained 30 analysts in TrueAllele, and they're very familiar with it. They know what the computer can do. So if you take 20 inconclusive cases from 1 week times 50 weeks, that's a rough estimate of 1,000 cases a year. Over 4 years, that's about 4,000 cases. And this area of New York, upstate New York, again has about 8 million people in it. So the country relative to that area is again about 40. Their estimate of 4,000 cases times a factor of 40 would be for the country maybe 160,000 inconclusive mixtures since the Swigdam rules. And as the analysts in those labs point out to me, we're not even talking about the underreporting of DNA match statistics. How 100 billion match statistic gets reported out as 100, or at that level. So the DNA evidence data in 100,000 cases that have been collected, analyzed, and paid for by taxpayers, that most of which our studies show are highly informative, that could be used to protect society, are thrown out. And they're just not used. For some people, find that a problem. I'd like to tell you about DNA databases before I end. The purpose of a DNA database is really a great idea. Imagine that you had lots of criminals. The FBI has profiles on about genotypes on about 11 million criminals. And you gather them as convicted offenders, maybe from the prisons, and you upload them to a big database, and they called CODIS, Combined DNA Index System. And this is kept at the national level called NDIS, which National DNA Index System, and maintained at the state level, correct, ESDIS, and so on. And now all of these DNAs of potential offenders, their genotypes are up on a database. Imagine that all crime scene evidence was single source. Well, that would be great. Then you could take from blood or very simple samples DNA from hundreds of thousands or millions of crime scenes, upload them to an evidence database. On the left side of this picture, you'd have the genotypes of evidence. And then automatically, using computers, these genotypes could be compared. And you'd be able to connect crime scenes with criminals through this database. And you read about that. And you hear about hits using CODIS almost every day. It's a great system. It's a phenomenal first start. But it doesn't work with mixtures very well. In fact, only about 10% of mixture evidence is ever uploaded to CODIS, which is interesting if you consider how many sexual assaults are mixtures. The reason is they don't use much math. It's all done by association. They don't have probabilities and genotypes. They have alleles, these lists of peaks that are under or over thresholds. And when they make comparisons, because it's being done by association, not by match statistics, they can make a lot of false hits once the data gets a little bit fuzzy, a little bit ambiguous with more alleles. It's not a very good representation. It loses tremendous information. And in order to prevent false hits, so the police aren't spending all their time arresting the wrong people, they restrict the upload to only the simplest mixtures with the simplest alleles that satisfy high match statistics for CPI that don't get that high anymore. And that way, they lose the true hits for mixtures, maybe about 90% of them. And so in this old style 20th century database, the criminals are up there, but the mixture evidence is not getting up to the database. It's not on CODIS. And I don't think it even comes up on TV, but you're not connecting mixture evidence, which is the prevalent form of evidence in most crime labs today that I know of, with criminals, because the pathway is broken and you can't get them up onto the database. And even if you did, they might match 10% of the criminals because it's using alleles and older methods. A TrueAllele database can upload all the mixture evidence, and that the associations that are made, the hits, are based on match statistics, based on real math. It's sensitive, as we saw in the study an hour and a half ago, and in many other studies, in that it finds the true hits. But the reason this architecture works is that it's specific. It doesn't find false hits. It really only finds true hits because the math is right. It's using the probability, the match statistics. On good crime lab data, what will happen is you can restore that pathway. All mixtures can be uploaded as their component genotypes as evidence. And because they're now up there and the comparisons that are being made are using real math, you're not overcalling and you're making hits. And so now you can connect crime scenes to criminals. One of our groups, one of the labs that's using TrueAllele, it's a small lab, but they're solving 200 mixture questions a day, and automatically evidence, criminals are being uploaded to their TrueAllele database, comparisons are being made. It so changed their workflow that when the lab director left for the American Academy meeting in Seattle last week, one of his analysts said, okay, we solved that sexual assault. And the lab director said, really, what'd you get a hit to? He said, I don't know, I haven't looked at it yet. I just put it in the computer, and the computer's already told me that that individual is a hit against this rape evidence. I'll look at it later. If you understand what goes in a crime lab to get to that point manually, and then throw out the information, as opposed to having all of this working automatically in a database, it's phenomenal. It's how a small lab with 4 or 5 people can handle a large county with 1 million people, because all the work they used to do is now being done by machine. And they're just harvesting the results that the math and computers are finding in the DNA evidence. So I think that's probably a good enough introduction for Becky Spanger, whom you all know.
She's first ADA of Allegheny County.
And I would just like to recognize that Dr. Perlin Williams from our laboratories is here as well, and there may be some other lab personnel here, but because we're interested and focused on this, getting the right person as early as possible in the system. We're currently working with Dr. Perlin's group at Cybergenetics, along with the Department of Laboratories, to put together a database study and start to use TrueAllele on an ongoing basis in our lab. They have the software. It hasn't been validated. We haven't gotten to that point yet, but we are working towards using this method in Allegheny County so that we can get twofold to 2 things: increased public safety and savings of taxpayer dollars. Public safety concerns we look at this as we want to put our law enforcement resources into looking at the right perpetrator. So if we can do with TrueAllele what we can't do now, which is exclude someone, we want to do that. We don't want someone to be under the cloud of suspicion wrongfully. We want to go forth and put our scarce law enforcement resources into catching the correct perpetrator. So doing this study will allow us to do that, move towards using TrueAllele. So we'll have increased public safety. The innocent will be excluded more quickly. It will work towards conviction integrity. We'll get better results when we have positive results, when we have an inclusion identifying the perpetrator, which gives us better conviction integrity, which I believe we're all looking for. And then the taxpayer savings is this. The software is currently in possession of the lab. They haven't been able to use it yet, but we're working to get to that point where they are working with this on a daily basis, which will save the analysts time. They'll be able to get to this more quickly. And instead of us having, on a select basis, having to write to the lab and say, send this particular evidence to Dr. Perlin's lab, have them analyze that on a case-by-case basis, and be paid a fee for that, and then have to be paid to come in and testify as an expert witness. Our lab personnel who are currently on the case panel will be able to do this on an ongoing basis, which will be a tremendous savings for the taxpayers of Allegheny County, and we'll be getting better evidence. And that's our goal, is to make sure that we are prosecuting the right people, that we're prosecuting them well, and we're doing it effectively and efficiently using the taxpayer dollars. We've been working with the lab, I personally have been working with them over the last couple of months. It's been an ongoing project for a couple of years. I've been in contact with legal counsel from both the FBI and the Pennsylvania State Police, because they govern some of these areas within the lab's control. And I've been in weekly contact with counsel from the FBI for about the last 4 weeks. They have told me that probably by next week they'll be able to give us a memorandum of understanding, whatever they want to call it, going forward, that they don't have any objection to the project. They're not saying that yet, but I've been told that I can expect to get something possibly by next week, that they have no objections to us going forward. To this point, they have given me no concrete objections, but I was told that this is what we're looking to do, is a novel project for them, so we're sort of cutting edge of what we're trying to do. And they're hopeful to be able to work with, they're encouraged and looking to work forward and move forward with this. Within the next week, we should have an answer from them. So that's our goal, that's what we're looking to do. And hopefully everyone, including those of you from the defense side of the bar, will see the value of this, because we will be excluding the innocent at an earlier time in the process.